To AOCS Webinar Wednesday. It is noon, so Phil, whenever you're ready, we can begin.
Thank you, Dan, and appreciate everyone taking time out of their day or their evening or their night, depending on where they are in the world, to join us. This is the next in an ongoing series of webinars around protein science and technology. That's all leading up to a very important meeting and to be held in the first week of November in Toronto. This will be the first annual Pulse Science and Technology Forum that is sponsored by AOCS. And so today we have a subject matter expert in the field. We think you're going to enjoy Dr. Pam Ismail's presentation on structural and functional properties of plant protein isolates and hydrolysates for various applications. Pam comes to us from the University of Minnesota's Plant Protein Innovation Center, where she is both founder and director. Pam has 2 decades of experience in various aspects of food chemistry. She's focused on everything from analytical chemistry and protein chemistry and actually fate of bioactive components in addition to proteins for a number of years. Its interplay between processing, fractionation, and those functional and bioactive attributes have been a focus of her interest and attention throughout her career. So we're looking forward to Pam's presentation today. As Dan has mentioned, we welcome any and all questions. Please use the chat function, and we will address them after Pam's had the opportunity to give her presentation. So without any further ado, Dr. Ismail, I turn it over to you.
Thank you, Jeff. Appreciate the opportunity to give a talk today or a webinar. Thank you, everybody, for joining the webinar today. And I'll walk you through a presentation that I gave earlier at the AOCS meeting with a couple of added slides, a little bit, to cover about 30 plus minutes, and then we open up for questions. So I will start up with something that you're not surprised about. It's the global demand for protein ingredients has been witnessing a steep increase, and it's estimated to reach about 7 million tons by 2025. Accompanied with that increase in production is obviously an increase in market revenue, which is expected to reach about $70 billion by 2025, and the rate of growth is about 8%, estimated between the year of 2014 up to 2025. Of this market, or of this global demand, 40% represents the protein, the plant protein market. So why, among the reasons that we see for the increase in plant protein interest is mostly consumer drive. So from a consumer perspective, there is a growing interest in the sustainability and environmentally friendly sources. There's also an increase in traction to plant-based and healthy diet, as well as seeking proteins that are non-allergenic due to the rising incidences of allergenicity of several of the key proteins in the market. Of course, from a producer perspective, the one thing that they aim for is addressing the consumer's demand. So if the consumer is asking for novel and new sources of plant protein, then the producer needs to address that demand. Another big thing for producers is finding a unique and a competitive space in the market. So how can they compete and find their own position in the market by introducing new and novel protein sources or plant protein sources? Also valorizing byproducts. So we are producing, for example, different oils, say canola oil. Same with soybean oil, you end up with a meal rich in protein. So how can we valorize that meal after oil production, or let's say after starch production as well? From different crops, you wind up with a material that goes to animal feed that is rich in protein. So how can we valorize such byproducts? That, not to mention the increase in population, or the population growth, which is estimated to be about 9 billion by 2050. So higher population means more food security is needed to feed the growing population, and discovering alternatives to animal proteins to satisfy the nutrition and need of proteins in general. So with that being said, there are challenges with novel and new and alternative proteins. Challenges are we don't know much about them since they're new. So we need to know how these new proteins that are gaining interest can replace or be combined in blending formulations with traditional protein ingredients in various food products, so that we can get the nutrition that is needed, the functionality, as well as maintaining good flavor or acceptable flavor. And with new sources of proteins comes with, okay, we need to extract this protein and develop or produce isolates. So we need to look into viable, cost-effective extraction and processing technologies for producing these functional protein ingredients from the novel sources. A third important one is you might find unique characteristics for these novel proteins. So we need to identify these unique characteristics or functionalities or nutrition and see how they can be used in different applications based on the protein source. So ultimately, we need to demonstrate either an equivalent or even better functionally active novel protein compared to the existing protein ingredients. So this is essential not only for the food industry, but also for consumer, especially when we're looking at nutrition as well as flavor. So many of you know the current trendy application, a big one that is gaining a lot of traction right now, is the meat analogs. So it is expected that the market will reach about 5.2 billion by 2020 and also make up 1/3 of the market by 2050. So this is a huge and not going away trend. Another trend that was witnessing increase in interest is the protein beverage market. And this market has been seeing a steady increase, and it's expected to be about 2 billion by 2025. Other than these two, there's always the protein bars market, which is also of interest and growing. Cheese analogs is another one, extruded products as well. So, but when we listen to industry and consumers, they're looking for ingredients to utilize in these products that are alternatives to soy and gluten. So it's not only plant protein that they're seeking, but also seeking alternative to soy and gluten for many reasons. Among them is soy protein coming from soybean GMO, genetically modified, and a lot of buzz around the health issues of gluten, and also the allergenicity accompanied by both soy protein and gluten. So peas, we all know that the pea protein is becoming a trendy alternative protein ingredient in the market. So looking at peas as a crop, it has several environmental benefits. One, first it's a short growing season, so that makes it easier to grow and produce and could be in a crop rotation. It fixes nitrogen in the soil, it enhances the nutrient levels, and especially when it's used in crop rotation. Other advantages, currently there's a lot of production that is non-GMO, And as of now, the occurrence of allergenicity is low. And I say this with a caution, and I say now, but any protein can potentially become allergenic, especially when there is an increased exposure. But as of now, the interest in pea protein is one of the main reasons. It's currently not on the big 8 other allergens, or the FDA big 8 allergens. So looking at market trend in terms of percentage of protein product launched using soy versus pea, we notice a decrease in the use of, in the percentage of protein product launched from soy, whereas we see a steady increase in the products from utilizing pea. So we see an increase from about 7.5 to about 21% in the market right now. So, and this is going up, but there are limitations hindering the growth of peas. So there are several things in the way of peas getting ahead of soy. So one big one, it's inferior functionality compared to soy protein. Soy protein had many years of research ahead of pea that looked at ways to functionalize the protein, and inherently it's a higher functional protein than pea protein itself. So if we look at an application which is an acidic beverage application, high protein acidic beverage application, that's why we look at solubility at pH 3.4, which is the pH, or approximately the pH of an acidic beverage. We look at the pea protein isolate, a commercial pea protein isolate. It has less than 10% solubility compared to soy protein. It has about 20%. Not that great compared to whey protein, which is the golden standard for beverage, but in any case, pea protein solubility is inferior to soy protein. Other than that, we have flavor issues still with the pea protein, given the different flavor compounds that can come from the crop itself and can be highlighted or produced during the extraction process, which again, soy protein manufacturing has years of research ahead of pea to mitigate flavor, whereas pea is just starting to look into that flavor issues. So we have, because of the inferior functionality and the flavor issues, we have an impact on the inclusion level. How much protein or pea protein can we add to our formulation, and also impact on shelf life in terms of not just flavor, but also if you are thinking of a beverage, there could be a potential of aggregation over time and a loss of dispersibility over time. So what we're doing is looking at ways to kind of give pea protein viability, or give pea protein a chance in comparison to soy. So in order to do that, we've looked at different ways to functionalize the protein, or to make it more functional and the structural integrity is preserved. We do that with looking at optimizing the extraction methodology. So there are common extraction methodologies that are industry utilized. So most significantly is the isoelectric precipitation method. And then there's the salt extraction and the ultrafiltration, which is very expensive, but it can be industry feasible. So what we're trying to do is looking at taking those traditional methodologies and optimize them to make them more efficient, enhance the yield of protein production, and maintaining structural integrity of the protein so that we can preserve some of that functionality. Another thing we're looking at for flavor in particular is we are developing flavor guided extraction. That means we pick out samples at different extraction points and look at the development of flavor or the loss of trouble flavor compounds and see how we can mitigate either the production or enhance the removal of these flavor compounds through changing the steps of extraction. Another area we're working on is functionalizing the pea protein after production. So we're looking at targeted enzymatic modification, Maillard-induced glycation, targeted fractionation, and most recently we are starting to look at the application of non-thermal processing such as the cold plasma. This project, or most of this project, is funded by USDA ARS. So just a quick a little bit of information on our progress in the optimization of extraction methodology. Looking at solubilization using the isoelectric point precipitation, and also looking at protein salt solubilization followed by ultrafiltration rather than precipitation by salt. So we want to eliminate the use of excessive salt, which makes it hard in terms of wastewater and dealing with salt in the processing plant. So by doing these two methods under optimized conditions, we looked at the structural integrity of these protein extracts. We did differential scanning calorimetry to look at the denaturation, state, and temperature. And from here, we can see that we have two endothermic peaks, one for viscellin and one for legumin in the two different proteins extracts. And that tells me that the protein is not being denatured in comparison to a thermogram of a commercial pea protein isolate where we do not see any endothermic peak, which is an indication that the protein has been denatured already. So another difference we see here is the use of salt extraction versus pH extraction. We see a change in the denaturation temperature where the denaturation temperature has shifted to a higher denaturation temperature for both viscellin and legumin in the salt extraction. So they are slightly more thermal stable than the pH extracted pea protein. So with that, looking at a complementary or confirmatory method, looking at surface hydrophobicity, and that's an indication of unfolding of the protein due to denaturation. We can see that with the commercial pea protein isolate, we have higher surface hydrophobicity, followed by our pH extracted sample, and the lower surface hydrophobicity is with our salt extracted sample. So looking at functionalization of the pea protein, I'll give you a little bit of results for the enzymatic modification, but just to introduce that a little bit, when you have a protein, you can hydrolyze the protein obviously by a wide range of enzymes. You have many enzymes to select from, and you have a wide range of pH, temperature, and time, but by controlling these conditions and selecting certain enzymes, you can produce peptides with unique functionalities. So you can optimize your enzymatic hydrolysis process to target a certain enhancement in functionality. So just as an illustration here, I just want to highlight that how we do that with enzymatic hydrolysis by selecting certain enzymes. In this example here, in lane D, you see that the enzyme we've used hydrolyzed the viscellin proteins, whereas legumins remained intact, whereas another enzyme we used hydrolyzed the legumin proteins and left the viscellin proteins intact. So we can potentially select enzymes based on their specificity to target several different subunits to result in profile of peptides with targeted enhancement in functionality. Another approach we've been looking at is the Maillard-induced glycation, which is based on the initial reaction of the Maillard reaction, basically. So you have the initial and then the intermediate reactions and then the advanced stage reactions of the Maillard reaction. But what we focus here on is just the initial reaction, which is the condensation reaction. and it's a reaction between the reducing end of a carbohydrate, a carbonyl group, with the amine group of a protein, and mostly a reactive amine group, such as the amine group of a lysine would be most reactive in this case, to generate an amidori compound, which is the first stable compound in the Maillard reaction. And that amidori compound in this case is a glycated protein. So what we do here, we control the reaction. So we have we conjugate or we glycate certain locations on the protein, not all possible lysine locations, because you don't want to lose the nutritional value of the protein either. So we do partial glycation that will result in steric hindrance and repulsion between the proteins to prevent protein-protein interactions and protein-protein aggregation. That will potentially enhance solubility. So I'm going to show you a model for enhancement or effect on protein structure upon glycation, and we did that in whey protein. And here it's a thermogram using a differential scanning calorimeter to look at the denaturation temperature of beta-lactoglobulin upon glycation. So we see a shift about 16 degrees shift in the denaturation temperature. That means an indication of enhancement in thermal stability upon glycation. With that, the complementary look at the surface hydrophobicity, where we see that the surface hydrophobicity of a non-glycated protein increases with heating time. So as you heat the protein, in this case at 75 degrees C, as you heat the protein, it's going to denature, unfold, reach a maximum surface hydrophobicity, and then it decreases due to polymerization with increased heating time. Whereas a glycated protein, we see a slight unfolding occurring and then plateauing over time of heating time. That's an indication that the protein resisted unfolding, unlike the non-modified protein with thermal treatment. So here is when a protein is denatured, it will potentially form polymers and aggregates due to hydrophobic interactions and disulfide linkages. So a protein that is not modified, we see here beta-lactoglobulin and alpha-lactalbumin. When you heat at 75 for 10 minutes, then you start seeing polymers formation and then reduction in the intensity of the monomers. And that polymer increases in intensity as heating time increases and monomers decrease in intensity. So that's an indication of polymerization due to denaturation, which leads to reduced solubility. Whereas a glycated protein with heating, you don't see the formation of polymers, and you see the monomers remain throughout the heating time. Looking here at Raman spectroscopy to look closer at the impact of pH and temperature on the structure of the protein when it's glycated and non-glycated. So at pH 3.4, around the isoelectric point of the protein, and at neutral pH. So this is a non-glycated protein not heated and heated versus a glycated protein that is non-heated and heated. So what you can see at the different pHs is the glycated protein does not change there's no change in the spectra. So there is no change in the protein structure. That is, when you heat the protein, that means it maintained its rigidity. Whereas the non-glycated protein or the control protein, you can see spectral changes, shifts, and changes in intensity of the different spectra or spectral peaks, indicating a change in the protein structure. So another way we're looking at functionalizing the protein is by targeted fractionation. So following unique chromatographic separation, we are able to get two fractions of the pea protein. One we call a water-soluble fraction, and the other is a water-insoluble fraction. And the hope with that is to produce two fractions for different applications such as extrusion that requires polymerization. We might tend to use the water-insoluble fraction. whereas the water-soluble fraction can be used in applications such as a beverage application. So here we look at the distribution of the protein bands of the water-soluble versus the water-insoluble. We can see that the water-soluble contains minimal, if any, polymers, whereas the water-insoluble fraction contains your high molecular weight polymers and some monomers as well, but mostly participating in protein-protein interactions. So looking at an impact of these different modifications and approaches on functionality, I'm highlighting protein solubility. So if we look at the commercial pea protein isolate, it has, as I showed you earlier, less than 10% protein solubility, whereas our modified, our isolates produced under optimized extraction conditions. So looking at the optimized isoelectric precipitation, we have increased solubility by about 4 times. With salt combined by membrane filtration, we increased it 6 times. When we optimized hydrolysis of the pea protein and we used the commercial pea protein and we hydrolyzed it under optimized conditions, we also saw a 6-fold increase. When we fractionated and took the water-soluble fraction, that water-soluble fraction had close to 90%. And if you see here with the glycated pea protein, it had 100% solubility. So that was our most successful product. So I'm going to move on to 2 different types of novel proteins from Camelina and pennycress. These are new crops that we're looking at, and they are oilseed crops, and they are from the Brassicaceae family. They are winter short season crops, and they have a multitude of environmental benefits, including reduced soil and water erosion, reduced nitrate leaching, increased carbon sequestration, and reduced inputs for energy and pesticides. And they can, because they're short growing season, they can fit in crop rotation between corn and soybean. So they are both, I see questions trickling in. I will definitely respond to those at the end of the presentation. So both of these crops are rich in oil or fat and also protein. And when we press out the oil, we end up with a cake or meal that is high in protein and fiber. Same as the case with soybean meal or cake. So that's another situation where you have protein-rich cake that we can benefit by valorizing this byproduct. So since they're new crop, not much is known about their protein. So we're looking first at optimizing their extraction methods, and it's really different than soybean, slightly different given the slightly different characteristic of the protein themselves being more soluble in alkaline. So if you try to solubilize this protein at pH 7.5 or 8, the same as what we do with soy, it won't work. You will have a very low yield. So we're working on looking at ways to solubilize the protein out of that complex matrix to enhance yield and while maintaining the structure and function of that protein. So we're looking at structure and function characterization. We're also looking at ways to modify or enhance the functionality. And also we're looking at, do we have a problem? I just saw that somebody cannot hear me. Do we have a problem? Jeff or Dan?
No, audio is good.
Okay.
I'll email that attendee or message that attendee. But yep, audio is coming through.
Okay, good, good. All right, so the other thing, since there are new crops and looking at newer proteins, we're looking at protein nutritional quality. We're doing PDCAS analysis on the protein that we extract under different methodologies and modify. And definitely a big thing is we're looking at the flavor, and I have a couple of slides on that as well. So these attempts are funded by General Mills, Minnesota Department of Agriculture, and our internal Healthy Foods, Healthy Lives Institute. So we start obviously by either cold press or hot press the seeds to produce our cake, which is still high in fat. So we do a hexane extraction to defat, and we end up with defatted camelina meal or defatted pennycress meal. So, and then we apply two different methods, again, pH extraction and salt extraction with and without membrane filtration. And then we apply our modification on the protein isolate from there, either by hydrolysis or by Maillard-induced glycation. And then obviously throughout, we look at structural, functional, quality, and flavor. The flavor is what we intend to do is, again, flavor guided extraction. So we're looking at flavor at the beginning within the seeds themselves, then as a meal, then during the different steps of extraction. So just a quick look at the camelina protein extracts. Looking at the different components, we see a striking difference between camelina, for example, and soy. I don't have a soy protein here in the gel, but I can tell you that the proteins in camelina and pennycress as well are much lower in molecular weight than soy protein. And another difference here is looking at extraction with pH versus soy. We see a gluten type polypeptide in the pH extraction, which is not present in salt extraction. That means this protein was not solubilized by salt. And then the main proteins are your cruciferin and napin. Cruciferin is the globulin, napin is the albumin proteins. And then by running non-reducing and reducing, we know that we have disulfide linkages of the polypeptides. Another, when we ran 2D gel, another observation is that most of the proteins or the polypeptides are basic. So you can see them more towards the alkaline pH scale. So, and that's why these types of proteins require high pH in order to solubilize them out of the matrix. Looking at their denaturation temperature, they do have high thermal stability. Looking at cruciferin and napin, they both denature above 95 degrees. But one thing that you can see here is with the pH extracted, we don't see an endothermic peak, so there's no denaturation temperature that we could record. And that's because we use high alkaline pH to extract the protein, which resulted in the denaturation of the structure of that protein. And that was illustrated by the higher surface hydrophobicity that we see from the pH extracted protein, whereas the salt extract protein has lower surface hydrophobicity, an indication of the protein being not as denatured as the pH extracted protein. Looking at comparing it to soy protein isolate, soy protein has higher surface hydrophobicity, but because soy protein also on the surface has higher charge as well, so it has higher activity on the surface, whether it's surface hydrophobicity or surface hydrophilicity. So looking at protein solubility, I just want to highlight the results at pH 3.4, where we see that the salt extracted protein had higher solubility than the pH extracted protein, which is not surprising after seeing the denaturation state of the pH extracted protein. And another observation is that the solubility at pH 3.4 was higher than soy protein isolate solubility at 3.4. So in a protein beverage, acidic protein beverage, there is a better chance for camelina protein compared to soy protein. Looking at emulsification, here we look at the emulsification capacity, or the milliliter, or the amount of oil that can be emulsified by a certain amount of protein. We see that the salt extracted protein had higher emulsification capacity than the pH extracted, and it had comparable emulsification capacity to soy protein. However, slightly lower stability, but comparable emulsification activity to soy protein isolate. In terms of foaming, it behaved, the Camelina salt extracted obviously behaved better than soy protein isolate and had similar stability to soy protein isolate. So there is a good potential for Camelina here in terms of functionality or replacing soy. However, in gelation, it didn't gel very well, neither the salt extracted or the pH extracted compared to soy protein. And that is not a surprise. Again, the lower molecular weight of these proteins is hindering the formation of a good, strong gel. They do hold water well, but the gel strength was not as great. So a little bit about flavor. So we're looking at pennycress. I'm showing you an example of pennycress, and our flavor chemist, Gary Runexis, has I have a quotation here. The flavor of pennycress is like a mix of pepper, mustard, and onion with a slight bitterness. So that's not very promising, is it? But what we are hoping is through extraction, we eliminate some of that flavor. But originally, we want to see what they are exactly, and how can, and where do we lose them in the process of extraction of that protein. So if we look specifically at pennycress, the glucosinolate is the main source of the flavor, and sinigrin is the main glucosinolate in pennycress. And it can, through different metabolic reactions, can produce different compounds that are flavor active compounds. So allyl isocyanate, allyl thionate, and a couple others. So some have the mustard, horseradish flavor, the garlic, the sulfurous, the mustard, and horseradish flavor as well. So what we are attempting to do is looking at these compounds and monitor them, like I said, from the seed themselves to the meal and cake, and then from there also to look at these flavors. Either are they still present in our protein extract, or has something else developed along the line of production. So here's an example of using GC, gas chromatography mass spectrometry, for the pennycress seeds, and looking at one of the flavor active compounds, which is the allyl isocyanate. So we have a standard and then the sample to confirm. Looking at the spectra, we confirm by comparing to a standard that this is the compound, but we don't just rely on that. We also run GC olfactory, MS, to sniff the peak as they come out from the GC and confirm associate the flavor with the compound as it leaves the column. So we confirm that this allyl isocyanate has the pungent mustard flavor. So this work is ongoing, and we are continuously looking at this compound and other compounds throughout the process of production of that protein. So I will sum up with some takeaways. Demand of plant protein is steep and long-term. It's not going away, it's increasing. But we saw that some plant proteins have challenges compared to current proteins in the market. However, from what we've been working on, there is room for improvement. And yet the research in this space is much needed, is ongoing to address the need, the consumers and the industry need, the challenges as well as the opportunities. With that, I'd like to acknowledge my funding sources, students, especially these 4 lovely ladies who worked on the different proteins I've showed today. And I will end by thank you guys and open up for questions.
Thank you, Pam. This is Phil. We have a couple of questions. The glycation portion of the presentation generated a number of them. And so let me scan back up to the top since there's several of them. Several questions about the basics of the process. So first and foremost, is it physical? Is it an enzymatic process? And this is from Highwell. Then secondly, Tom asked, can you choose specific glycation sites or functionalities, either by choosing different sugars? And will these have any effect on either the taste for either meat alternatives, dairy alternatives? You could probably ask the same question about functionality. So the degree to which this process might be able to be controlled, either from a research perspective or a commercial perspective. Any thoughts or comments there?
Yeah, sure. I'll start off with whether it's physical or enzymatic. So it's not enzymatic. So we do the glycation via conditions that will induce Maillard reaction. So basically it's temperature, water activity controlled. So we have humidity controlled chambers that we kind of induce this reaction. However, we choose our carbohydrates in a way that they are not highly reducing. So it's not high reducing power, because if we have carbohydrates that are of high reducing power, then the reaction will proceed super fast and we cannot control it and stop it before advanced glycation products. We do not want browning. We do not want the advanced glycation product that can be harmful in terms of toxicity or in terms of even polymerizing the protein. So we control our conditions to a water activity just below the optimum of the Maillard reaction optimum. And we also control, like I said, the type of carbohydrate, that it doesn't have a high reducing power. And we also control the combination, how much protein and how much carbohydrate we are adding. So it is a physical process. It is, however, it can be done under aqueous conditions as well. We do it under dry with a controlled relative humidity chamber, but it can be done under a wet condition, but that could be also tricky because it can, the reaction can go very fast. So it needs more tighter control, I would say. And then to answer whether we can target certain sites, well, it depends on the protein, and the most reactive amino acid in the protein is our lysine. So it depends on the location of the lysine within the protein, and it also depends on whether the protein or not has open structure or has globular structure. And also it depends on the size of the carbohydrate and how easy, the bigger the carbohydrate, the more hindrance we have where it can access those lysine sites. So it's a very complex process, and it's very dependent on the type of protein. So one condition or one solution that will not work for all proteins. So it really has to be optimized for the protein and the source of protein and the denaturation state and the structure of that protein.
Very good. Can you comment as to?
I was finish up with the taste impact, because part of the question also asked about taste. So depending on the carbohydrate that you use, if the carbohydrate that you use doesn't have really a taste, and if you do not drive the reaction forward to advanced glycation product, we don't expect to see changes in flavor. I can't say 100% because we did not do flavor analysis on these products, but from the chemistry of it, and depending on the flavor of the protein that we're starting with and the flavor of the carbohydrate, and if the reaction is truly controlled, theoretically we shouldn't see a change in taste or flavor.
Okay, very good. Thank you, Pam. Follow-up question there. As an academic scientist, but one that collaborates with industry and government, what are you seeing in terms of startup companies or areas where people are trying to take these principles and turn them into commercial technology or commercial products?
So are you asking about feasibility, or are you asking about labeling, legality? What is it exactly?
Well, scale up feasibility, and then of course regulatory implications. You mentioned, for instance, if you have too much glycation, there may be either poor digestibility or even toxicity. So I'm wondering if you could elaborate on those comments that you made for the audience.
Yeah. Again, I want to emphasize that the procedure that I'm talking about is not result in advanced stages of advanced glycation stages. So there is no toxicity concern, because we do test the stages where we are at with the glycation. We do test that to ensure that we don't get to advanced glycation product. In terms of digestibility, actually our studies showed that partial glycation result in partial unfolding, and it does actually enhance the digestibility of the protein, not by a lot, but at least it does not decrease it, because it's partial glycation. If you glycate excessively, you can end up with decreased digestibility. So that's another thing. Now, in terms of feasibility, we have not yet done a scaled up testing. These were all lab testing. I'm looking to partner actually with a company that's interested to scale up, to do some pilot scaling up and determine the feasibility on a large scale. I haven't done those studies yet, but I would like to partner with somebody to do these studies.
Okay, so the contact details are here on your last slide, and this deck is going to be made available to the audience. So those that might be interested in that conversation, you'll have Pam's contact details here. A little bit moving towards Camelina and pennycress, since we don't see a lot of questions here. Let me pose a couple for you. The color of the press cake is obviously rather dark. You go through a subsequent hexane extraction, followed by a number of different extraction procedures. Can you comment on what the color is in the final product, and whether any of that affects your thinking of how they might best be used in an application where, say, color is critical?
Yes. Definitely. I can for sure tell you there is a color difference based on the extraction methodology. So when we do alkaline extraction, especially for Camelina and pennycress, because the alkaline treatment is really high, we end up with a really dark color, and that is because of the oxidation under alkaline conditions that is happening. You have the high phenolic group, you have the protein and pH, then you end up with a really dark color. So when we do the salt extraction, we actually end up with a color very light, similar to pea protein or soy protein isolate kind of color. So yeah, so in terms of extracting these proteins, I guess the pH extraction resulted in really denaturation, not so attractive color. So what we're trying to do is optimize the salt extraction to minimize the amount of salt used to produce these isolates with preserved structure and function, as well as color and taste and flavor. So.
Very good. Very good. You showed some second follow-up, and maybe this can spur some additional questions from the audience. You showed some interesting functional characterizations and comparisons between the Camelina and pennycress proteins and say soy protein, for example. One of those related to both emulsification, potential for foaming, functionality, foam stability. Given the given the role of textured proteins these days in various applications, either in meat alternatives or in nutritional bars, have you ever looked at Camelina or pennycress proteins for their performance in, say, an extruded application where foam functionality would play a potential role?
Yes, that's a great question. We haven't yet. We are at the very early stages of looking at these proteins, and we are doing our basic characterization at this point, looking at, first of all, how to optimize extraction, how to minimize flavor and color, and what kind of basic functionalities do these proteins have. We haven't yet gone into applications, and this is the next step. Definitely, we want to look at beverage application, extrusion application, bar, nutrition bar. We did a little bit of bars for Camelina protein. We didn't do an extensive storage study to see how the protein behave. Does it harden over time like soy protein does? We haven't gone into that yet, but this is something in the pipeline as well. Yeah, we are at the beginning of our research in this area.
Very good. Well, I'm not seeing any, well, one final question here then. Question on salt concentration. What did you use and which salts or salts have you tried in the salt fractionation or salt purification?
Yeah, so for salt, yeah, I see that question. So for salt, we use, to solubilize the protein, we use NaCl, and we are trying concentrations that range from 0.1 to 0.5%. And we are in the process of trying to optimize the best concentration based on the yield. And based on, we're doing pilot plant studies right now, not just our bench study, we're doing a pilot plant production to see how efficient we can use the ultrafiltration to remove the salt with the minimum amount of water needed. So this is still ongoing. We have not yet gotten the final results to say, okay, we're going to go with 0.3% salt and do ultrafiltration. So we're still looking at the ash content in the final product. We're still looking at the efficiency of the membrane filtration, the fouling, all of that. So this is still in process.
Well, lots to stay tuned for. Very good, very good. So, Pam, I'll say on behalf of the audience, appreciate the time and effort to put both this presentation together, and as well the effort that you put out to both found and lead the Innovation Center there at the University of Minnesota is I think one of the keys to this very exciting landscape around plant proteins. And we look forward to future engagements and other contributions to the literature from you and the team there and the center as a whole.
Thank you so much.
On behalf of AOCS, thanks for your time here today.
Thank you so much. Thanks everybody for being here today.
Dan, any last comments?
No, that's it. Thank you, Phil and Pam. Everyone have a good day.
Thank you. Bye-bye.
All right. All the best.